All right, the second last video for Year 12 biology, and we're going to be looking at patterns or modes of speciation in this one. We've got three that we're going to look at, and I will just give you a heads up. There's a fourth type of speciation that you may become aware of in some other sources, but we aren't required to learn it according to the QCEE syllabus, and it's also a bit of a sort of hand-wavy new one known as peripatric. But in today's video, we're going to be looking at allopatric, parapatric, and sympatric speciation. And we're also going to be looking at how they can sort of be seen as basically a spectrum, even though we will be looking at them separately. Now, as we know, speciation occurs when organisms become reproductively isolated, and then microevolution occurs to such a degree that we end up with two distinct species. Now, in order for this to happen, generally, we need a throttle on gene flow. And allopatric speciation, our first type here, has zero gene flow between our subpopulations. Parapatric speciation has very low to no gene flow, and sympatric speciation occurs in circumstances where we do still have high opportunity for gene flow, and we'll talk about how that happens later on. But just looking at this speciation continuum, we can see on one end we've got weak or no divergence, and looking at our genetic or phenotypic clustering there, you can see that these two populations are relatively similar, and they have a lot of interbreeding and a lot of gene flow between them in that sort of weak end of the spectrum. As we start to move further along, you can see that there's less interbreeding as indicated by less of those double-ended arrows, and they start to show greater divergence from each other. And then on the end, we no longer have interbreeding between those top black circles and the bottom white circles, and at this point they are so genetically different and they've had such strong divergence that they are at this point different species. So this just reinforces what we've been talking about previously in regards to microevolutionary changes leading to speciation and then macroevolution. So looking at our first one here, allopatric speciation, that's occurring when gene flow is completely disrupted. We have zero gene flow due to physical or geographical isolation. So two populations have been completely cut off from each other. As you can see in that diagram, some sort of barrier has been formed that completely isolates them. And because of this reduction in gene flow, different selection pressures or random genetic drift are going to act on these two populations, causing them to undergo microevolution, diverge from each other genetically, becoming more genetically different over time, and then resulting in speciation. So there's absolutely no mating or gene flow in these populations, and this is the most common form of speciation, and it's probably what most of you think of when you think of evolution and speciation. It's definitely the one favored most throughout evolutionary biology. So species can be separated, resulting in allopatric speciation in a variety of different ways. They can be separated by water, so flooding or changes in river patterns or increasing sea levels, that sort of thing. And be separated by massive tectonic changes and things like canyons, earthquakes opening up, fissures in the ground, moving of islands through continental drift and the like. They could be separated by land all of a sudden forming. If you've got a population that lives in an estuary sort of situation, they can be separated by sandbars with changing sea levels. Rising sea levels we've already sort of talked about, mountains and all those massive tectonic changes. So anything that's a physical or geographical barrier is going to effectively separate populations, resulting in allopatric speciation. Climate change also being another major one. This is a fantastic example here, again, going back to the Galapagos Islands, of allopatric speciation, because we do often see it in islands, because islands, generally were attached to the mainland or to other islands at some point and separate off. But Darwin observed this species of flightless cormorant in the Galapagos Islands, P. harrisi, which it's believed likely originated from an ancestral flying species of cormorant, which still exists on the South American mainland, P. brasilianus. So it's theorized that at some point an ancestor, one of these organisms that was, the ancestor of both of these species, flew to the islands. And as a result of that physical distance and, maybe not flying back, we had allopatric speciation was allowed to take place. Now, allopatric speciation doesn't just occur. We do need some sort of change in our selection of different traits. And what happened was there was no predation on the Galapagos Islands. There were no natural predators for the cormorant. And so as a result, they didn't rely so much on being able to fly because they didn't need to be able to escape from predators. Whereas the flying species in the Amazon would have to worry about escaping from caimans or jaguars and whatnot. So as a result, the most favored trait, the one that made them fitter, was being able to move in the water. So they reduced the size of their wings, and they changed their body shape somewhat so that they are better at swimming. So they were responding to two different lots of selection pressures, and as a result, we end up with a different species on the Galapagos Island that is flightless, and one on the South American mainland that can fly. So that's a great example of our allopatric speciation there. A lot of this can also occur through habitat fragmentation, when we have areas that are cleared for agricultural farmland, when we have mining that's tearing huge scars in the landscape, or when we have things like roads, highways, they can separate and fragment natural habitats. As well as this, other natural processes such as flooding or droughts or earthquakes can separate those habitats into smaller, more isolated fragments of those habitats. And as a result of that, there's less gene flow. If you look at this image here, when that highway got put in, it would be very difficult for a lot of species that rely on land to be able to cross that highway without getting hit. So as a result, we had one population that lived on throughout that entire forest. They've now been isolated thanks to this highway. Now, to overcome this and to prevent having them completely physically isolated, you can put in wildlife corridors and little land bridges and things like that there that can prevent the fragmentation. But when that fragmentation does occur, and if selection pressures are somewhat different on the other sides of the fragments, then allopatric speciation can occur. It's one of the reasons why wildlife corridors are so important. I'll skip that for now. Sympatric speciation is our second type, and it's on the far end of the spectrum. It occurs without any physical or geographic barrier, and it is when we have a sort of reproductive. We don't have reproductive barriers to gene flow in the same way of allopatric speciation. We still have high capabilities of gene flow, but they split into sort of different subgroups within the population based on things like feeding preference or mating preferences, temporal isolation. So we still have reproductive isolation, just not in the same sense as allopatric speciation. And due to these reproductive isolating barriers, whether it be behavioral, temporal, then the two populations are prevented from actually, true gene flow is prevented, even though the possibility of gene flow is still high. And there will be a lot more, interbreeding in this circumstance because it's available. But typically these populations will separate. As a result of this, because we've got no strict separation and because gene flow is still possible, then these sympatric speciation events tend to take much longer times. And then, as with any speciation, if different selective pressures act on these two populations that have separated themselves from each other, allelic frequencies will change and speciation will occur. So this is occurring without geographical or physical barriers. Instead, it's behavioral or temporal barriers. And this is a fantastic example here of some fish, okay? We've got these, as you can see, these are all part of the same genus, but different species. And this is in Central America. All of these four fish are very closely related, even though they may look somewhat different. Some geographic isolation has occurred in these fish, which has resulted in allopatric speciation from the common ancestor, which was A. citronellus. Okay, and you can see that A. citronellus is highlighted in the red there. Okay, so we've had some geographic isolation, say, between that Lake Apoyo and the other lakes. As well as that, some fish of this genus have chosen to live on the bottom, so they're benthic living fish, and that's where they feed, and others have chosen to live within the water column or close to the top, being limnetic. As a result of these different behaviors, if you're hanging out on the top, you're not going to reach the bottoms, and so, tops and bottoms aren't going to interact in this case. And as a result, they're only going to be mating with other tops or other bottoms, and that results in sympatric speciation. So in this case, we've had both allopatric speciation sort of initially kicking things off, and then followed by sympatric speciation. So the allopatric speciation would have given the Lake Ixolia original ancestor and the Lake Apoyo original ancestor as they were separated, but then through sympatric speciation, we ended up with the two different species in each of those lakes. This brings us to our last form of speciation, parapatric speciation. Parapatric speciation is in the middle. We have a low gene flow, but generally almost none. And it happens when the gene flow is not disrupted by physical or geographical isolation, but it forms sort of an adjacent niche. It's right next to it. So they sort of just stop living near each other. And generally this is due to some sort of extreme change in the habitat. This type of speciation is quite rare because they are so close to each other. Okay, so gene flow along the bordering areas will occur between those two adjacent niches, but it will become more and more reduced over time. Okay, so this is occurring through not strict physical or geographical isolation, and it's not necessarily through behavioral or temporal either. An example of this is when it comes to mining waste contaminating the soil. Okay, so the contamination will be toxic to most plants, but some of those grasses might just be naturally tolerant to those contaminants. They might have a mutation that then allows them to grow in that area. So they can then grow in the mine waste, and because they don't have to compete with the other grasses that don't have this mutation, then they're going to happily live in that mine waste, forming an adjacent niche. You can have some gene flow because at this point they're not completely isolated from each other reproductively, but over time they will become more isolated from each other. So there's minor gene flow between these populations. When we look at the three types of speciation, there is some sort of disgruntled evolutionary biologists out there who believe that sympatric speciation is rare and difficult for it to occur due to the lack of that geographic isolation. There's a lot of scientists out there who think that geographic isolation is basically a prerequisite for a lot of this speciation. And you can see the scale here looking at the difference in terms of how separate populations are in our three types of speciation. Now, we have observed all three of these types of speciation, but by far, allopatric speciation is the most common. You see here, there's a famous example conducted in a lab of artificial speciation, looking at fruit flies, our good friends Drosophila melanogaster, which are always experimented on for genetics and evolution, and they separated them into two different populations. So I suppose it's sort of like an example of parapatric speciation in that they were in adjacent niches where the starch medium and a maltose medium. After several generations, they put them back together and they found that the starch flies only really enjoyed hanging out with other starch flies. And the maltose flies really only hung out with the other maltose flies. Maybe they only really hooked up when they were near food or something. I don't know. So as a result, they started to separate each other from each other in terms of those particular traits. This is artificial speciation, though, not natural. All right, to wrap it up, you've got a summary question there, so have a crack at it. If you are able to do this, then you are all over this syllabus dot point. All right, one more video left, and then we're all wrapped up.